Okay, thank you very much. We'll get this second session of the day started. And it's a very great pleasure to introduce Professor Wim van der Poel from the Netherlands, who is, Wim, I think it was about 10 years ago I first heard you talk, or maybe longer actually, maybe even 15, when people hadn't really heard about One Health, and you were one of the first people who was talking about it. And you came to Liverpool and educated us about it. So it's great that you're here and talking about One Health still and the COVRIN-project. Thank you very much.
Yeah, thank you very much. I was asked to present about the COVRIN-project, especially focus on outcomes of the COVRIN-project. I'll start by explaining what the COVRIN-project was, and I'll try to zoom in on the results of the project, because the project ended beginning this year. And I cannot present everything that was done on the research in the project. That's really impossible, because it was a large project and there were a lot of partners involved. But I'll try to give you a flavor, and I'll try also to make some side steps on research that we have done in relation to work in the project. The COVRIN-project was a project within the European Joint Program for One Health. That's a program that is ending the end of this year. Actually, we had the final meeting beginning this week in Paris. And in the European Joint Program for One Health, there are a lot of internal projects and also PSD projects. And the COVRIN-project was one of those internal projects. And I led the COVRIN-project together with Professor Daniel Horton of the University of Surrey. And there were a lot of partners involved, but I'll come to that. The European Joint Program for One Health had more than 40 partners across Europe. We tried to include partners from every member state. That we didn't really succeed in that, because for some member states, the counter financing was really difficult. Because it was partly financed by the European Union and for a part also by the national government. But the UK, for example, and the Netherlands were heavily involved in the European Joint Program for One Health. And we were very active in integrating research, but also in collaborating on One Health research, especially focusing on zoonosis and AMR, and especially also on foodborne zoonosis. In the project, there are a number of work packages. I have circled here education and training, because I was deputy of the work package on education and training. But there were also work packages like 3 and 4 on the internal projects. I will not go into detail too much, but just to give you an impression on how the program worked. In work package 3, we had joint scientific projects. It was led by work package 3, and in work package 4, the joint integrative project. Integration of partners working together, but also integration of research was a goal of the project, and especially research using One Health approaches. In the presentation, I will say some about the structure of the COVRIN-project, the internal project. It was a joint integrative project, but there was a lot of research involved, because all the partners were already active on coronavirus research. So they really tried to work together in the COVRIN-project to make it more effective and to not duplicate research that was done elsewhere. The integrative goals of the project, the strategy of the project, and then on the key outputs and some concluding remarks. The operational objectives of the COVRIN-project were mainly 2 large integrative research objectives: to identify drivers of coronavirus emergence, especially SARS-CoV-2, and to generate data and build models to do risk assessments for SARS coronavirus 2, and also extending that to other coronaviruses. This is the structure of the project. I already showed the work packages in the joint program, and here we see the work packages in the COVRIN-project. All the work packages the research work packages and also the risk assessment projects work package using the data of those of that research are coming together in work package 4 to try to improve preparedness for coronavirus and especially focusing on SARS coronavirus. Outputs of the work packages. In the work package 0, we had the coordination and communication. In that work package, an important output was that we were able to look into what was or do scoping of all the research in coronavirus that was ongoing in Europe, and that was a lot. And the idea was by doing that we were able to show to the European Commission that we were playing an active role not to duplicate research and to improve collaboration of research. I will not go into too many examples of that. I will jump to work package 1, where we focused on SARS coronavirus detection. And for detection, we focused both on antigen detection and antibody detection. And we did a harmonization of genome detection, and we evaluated serological assays and also did harmonization of antibody detection. So we did interlaboratory trials with a number of partners to compare those tests and also to improve those tests. This is an example, antigen testing for partner laboratories, PCR, but also immunostaining, and also the rapid antigen test, as you probably all know. And conclusion of that work was that, the tests are good, but there is also room for improvement. And that another conclusion was that tests for, let's say, high levels of antibodies or higher levels of antigen testing was not a problem at all, but there was still room for improvement. And in the project, we were able to stipulate on some of those aspects. A side step of that is some work we did. I think that this is interesting, and I would like to explain a little bit. It's a published paper in Biology Open. We used bees in our institute to detect SARS coronavirus infected samples. And this is a picture of the work we did. This is the fixation of bees to use them for detection. And what we did is we presented positive swabs, SARS coronavirus infected samples to the bees. And we could train the bees by extending their proboscis to show whether this was a positive or a negative sample. We trained them using positive samples and sugar water. And by training those bees, we were able to show that bees can be trained to detect SARS coronavirus positive samples. We did, this is a picture. You can see the extension of the proboscis of the animal, then there's a positive sample. And that worked very well. And this picture shows, and it's a published paper in Biology Open, you can see it. We got a lot of publicity over this paper. You can see that in this graphs that the bees have a good learning curve, and there's also a good memory detention. And if you are interested, please follow the paper. We try to follow this up with work on volatiles that the bees really can detect, because it's clear that the bees do not detect the virus. They detect the volatiles associated with virus infection. For the samples, we used samples. We did use some human samples, but most of the samples were samples that we collected from the outbreaks in the mink farms. Conclusion for what I already said, sensitivities of the test could be improved and standardization could be improved, so partners were able to make some steps forward on that. Another point that I would like to say some words about is the detection of SARS coronavirus in meat processing plants. There was quite some, we didn't really do this kind of work in the COVRIN project, but I put in the slide because there was a lot of discussion about it. The European Union didn't really want us to work on SARS coronavirus 2 in foods because they were afraid that we would detect the virus somewhere and that it would give problems for trade. But I think that's a bit of a strange reaction. I think we should have done this, or should have done more of this research, because it was shown that in meat processing farms there was a much higher level of SARS coronavirus infection compared to other populations. So I think this should be part of our, should have been part of our work, and other workers have shown, other researchers have shown that foods can be implicated. We also studied in the project stability of SARS coronavirus, and in that part of the work, partners were able to show that the stability of SARS coronavirus 2 really depends on the type of surface and also on the sample type or the environment that SARS coronavirus is in. In protein-rich environments, the virus is much more stable than in other environments. And so we have a number of publications showing differences in stability in different environments. We also did that in the mink manure, because there was a question after the mink outbreak: when you bring the manure to the land, would that be a risk for spread of the virus? And in that work, we were able to show that the virus, when you culture the virus from the mink manure, we did spiking studies in mink manure, and by spiking the manure, we were able to show that when you look at virus isolations, the virus is not very stable. You're not, the recovery is very low already after half a day. But the RNA is very, very stable. So you can, by PCR, you can detect it for a long time. But when you do virus isolation, you can only detect it for 1 day or even less than 1 a day. So that's an important observation. That was reported to the governments to give them a risk assessment of manure in the country. This is showing the level of stability of RNA over time in manure of minks. In work package 4, we did a lot of characterization work. We worked on an algorithm for novel virus assessment. We did harmonized sampling methods. We exchanged protocols. We established in vitro and in vivo characterization methods. And we also developed a catalog for animal models. And to give you some impression, this is in vivo and in vitro characterization that was done in cell lines. Neutralization assays were developed and exchanged. But we also did ex vivo organ cultures, and we developed an air-liquid interface model to start to characterize SARS coronavirus 2. A number of these studies have been published and have really contributed to characterization of the different SARS coronaviruses detected, especially in animals. We also did a model evaluation of SARS coronavirus 2. We compared studies, and we did studies with different models. And from that work, it was clear that hamsters are a really interesting model. And we are still using them in our institute, and especially because of the fact that hamsters do show pathology, but also do excrete the virus, and so you can also do transmission studies, clinical studies, as well as transmission studies in hamsters, and it's an easy to handle animal model. So hamsters, from the work of our partners, were very interested. Hamster model is a very interesting model, and also the readouts. There's more and more information on the readouts in hamsters, so it's really improving as a model for SARS coronavirus 2 studies, and probably also, or potentially also, for other coronaviruses. We did studies in a number of animals. This is a study in cats. This is pathology in cats, and in cats you see also infections of course coronavirus like in humans, but the pathology is not very severe. So in cats you see only minor disease due to SARS coronavirus 2, but there is clearly infection. And we did in our institute we did a transmission study, and in that transmission study we showed, and this is also a published paper, and the interesting from this transmission study was that we could show that also through the environment the virus can be transmitted. So we put cats in a cage, took them out of the cage, and put susceptible cats in that cage that was left by the animals, and we could show that the newly introduced animals were infected by SARS coronavirus 2 through the environment. So that's an important observation also. I will not go into details, but this is also a published paper. The first author is Nora Gerhards. On work package 3, surveillance and risk assessment, we developed surveillance strategies. We did model studies. We did studies of risk factors, and we made a list of susceptible hosts. I will show some more of that later on. And we identified transmission characteristics of the virus, especially in the mink farms. This is from the mink outbreak. Pathology is like in humans. Case fatality rate is not very high. Their impression was that case fatality was very high, but in the mink farms, case fatality was about 4%. So certainly not all minks will die of SARS coronavirus infection. In fact, that is more or less comparable to what we see in humans. In the mink farms in the Netherlands, we had 4 clusters of outbreaks. And so we think the assumption is that there were 4 or 5 introductions of SARS coronavirus 2 in the mink farms. And between the farms, there was spread of the virus. And studies are still going on to see how these transmissions between farms occurred. Well, this was a paper that we published in Science. Here we showed that the virus was introduced in the mink farms by humans, but was infected back to people working in the mink farms. In the mink farms, about 50% of the people who worked in the mink farms were infected or were carrying the same sequence as found in the minks. So there was a very high level of PCR positives in the workers, and we also identified identical sequences from mink and the workers. So it was clear that these people the virus had been introduced by people, but also people were infected from minks. And that's the reason why we in the Netherlands called all the minks. In the Netherlands, that was not so difficult because we sort of accelerated a decision by Parliament that mink farming had to be stopped in 2024. And now it was stopped in 2021 because of SARS coronavirus 2. So mink farming in the Netherlands is not coming back. We are still studying this, as you already said. This is a time-calibrated maximum likelihood phylogeny to show or to shed more light on the transmissions between farms. We are related to this likelihood phylogeny. We are looking into the different factors like the workers, transports, and this is a paper that is in draft but will be published soon. But I can tell you that the workers seem to be the main transmission mode between the farms. We also worked on susceptibility of animals. This is work that is done together with EFSA. There is an EFSA report that was based for a large part on the work done by the partners in the COVRIN-project, looking at susceptibility, which animals show clinical signs, which animals replicate the virus, and which animals excrete the virus. And that's put in an EFSA report that's available online now. The final work package was on preparedness, looking at sample types. What are good sample types for surveillance? We have ongoing studies on phylogeny. Screening in different animals. We screening and surveillance a lot of different species. A lot of work is also done in bats in the project. I already discussed the in vitro and in vivo models. Circulation and transmission in cats is important. We looked at catteries also. The virus can readily spread in catteries, and we should see cats as a transmission, as a partner of the family, as a member of the family in transmission of SARS coronavirus. And we are still looking at farm-related factors and risk assessments. This is about the samples, evaluation of what are good samples for coronaviruses. Not only what samples are tested positive or are often tested positive, but also what samples can easily be obtained in different situations. So that's a little bit looking to the future when we would want to look at SARS coronavirus infections in general. What samples can we use? Not only animal samples, but also environmental samples. There are also discussions still on animals about their transmission option or hotspots. Ourselves in my institute, together with other Dutch partners, we did studies in pigs, and we showed that in some pigs we see replication, but we don't see excretion of the virus. But in other animals, there are different reports. Some research report replication, excretion, and others, for example, in rabbits, there are reports on replication and excretion of SARS coronavirus 2, and others report that rabbits do not really play a role in, or cannot really play a role in the epidemiology of the virus. But as we all know, in mice, we've seen different reports depending on the different new variants of SARS coronavirus. So this is ongoing. I think this should be ongoing work. Looking at susceptibilities with the new variants that we are going to see of coronaviruses. So that's where I would like to end. I hope this gave you an impression of the work that we've done in the COVRIN-project. I really must thank all the partners. I have not made a list, that's really too much. But I think we have done quite a lot of efforts to generate good research and to start good collaborations. Thank you very much.
Thank you, Wim. That was a really, really interesting talk. I now want to know more about bees, I think. So I think Miles has got a question.
Great. No, that was a lovely overview. Couple of short questions, if I'm be so cheeky. One is that have you ever looked at the mechanism behind the pathogenicity differential between ferret that we worked on was at Porton Down, such a mild infection, and to the mink? I mean, you know, 4% is still much more severe than what you ever see in ferrets. We never saw any severity, let alone death. And also, you're looking at, this was done about risk assessment to impact on policy within the EU. Do you want to comment on the Danish government's decision to allow reinitiation of mink farming?
Yeah, well, the first question, we didn't really do studies on that. I know that one of our partners, Erasmus University in Rotterdam, did work on that. We have not really done more, also not in the COVRIN-project. Other partners, I don't think other partners have done more to develop. I do know that the APHA did some work on mink, but I don't think there are other partners that did much work on mink following the experiences that we had with ferrets and mink. So I can't really answer that. And the second question, in the Netherlands, the decision was easier, as I said, because there was also already a decision by Parliament that mink farming should be stopped because of welfare reasons. Looking at the decision of the Danish government, where mink farming is much more important than in our country, I would not really support that because, as well, that was also shown by Diana, that minks can be transmitters of other viruses also, and building those large farms with so many animals you're sort of taking a risk that the virus will be introduced and reinfect humans. So I would not really be in favor, even apart from the welfare reasons, because in my opinion, we should not keep animals, or at least not so many animals, for furs.
Thank you. Julian? We need some ECRs to ask questions, not just Miles and Julian.
Shall I speak in a higher voice, James? Would that help?
If you want, you can.
Wim, this is just a completely trivial question from my knowledge, right? Are there any other viruses that have such a broad host range as SARS-CoV-2? Or is it just because we've spent so much money working on SARS-CoV-2 that we found these? So it's kind of an artificial thing rather than an actual thing.
I think there are other viruses that have a very wide host range. And I think, but what I think is very important here is that that these host ranges change. What we've seen in coronaviruses, not only SARS, but over the last 10 years, we've seen several coronaviruses popping up in different animal species, including humans. We have seen PEDV, we've seen other coronaviruses in pigs, we've seen MERS, and now we see SARS. 10 years ago, coronaviruses were not seen as a really important zoonotic virus, but now it's completely another world. And so I think that's the main lesson that we learn, that these viruses change host range, these viruses change and host susceptibilities change, especially looking at the changing biodiversity. So that's a lesson we have to learn, that by changing biodiversity, by changing our habits, we may increase the risk of virus emergence. So I would really be an advocate for looking into that and try to prevent this kind of development.
Simon?
In one of your slides, you described great variability between of the outcome of infection experiments in different species. What do you think about the reason for those differences with regards to the quality of the virus stock that was used, the dose and route that was administered, or whether there was differences between the colonies that were used? Yeah,
I think these different routes of infection, the different strains that are used, all play a role in the difference that we observe. So looking into those studies, you really should, if you really want to compare that, you really have to look closely in what was done and how it was done. And also the different animal species may differ. And also in vitro studies, we see differences in cell lines also. So it's very important to really include that in your assessments. And what was very important there during the SARS coronavirus too, I don't know if you've been part of that, but the discussions that we had in the WHO working group on animal experiments, we discussed this kind of differences that we've seen in the different studies. So I think one of that's the main achievement also of the SARS coronavirus too, that we start having these discussion platforms on what people are doing, what kind of research. And I think that's a very good one hell of achievement that we have created that kind of platforms to discuss the different observations and to really look into the different ways that people have used to do their experiments.
Yeah, I think that's a really valuable point, and a number of us were members of that group. So yeah, great. Thank you very much, Wim, for a fascinating talk and fascinating discussion. Thank you.